Thank you all for joining this morning. We're really looking forward to sharing this update with you. Just a few housekeeping things before we get started. The meeting is being recorded. So if you can, please keep your mics on mute just until the question and answer session. And then, you can raise your hand or put questions in the chat. Either way, whatever works for you. And then there's also a transcript feature if you would like that option as well. And I think that's it from my end. So I will send it over to Terry.
Thanks, Jenny. Well, on behalf of the National MPS Society and the International Society for Mastocytosis and Related Diseases, ISMRD, we're convening a meeting to talk about some exciting research that we're moving forward in terms of support. I'm going to first introduce Dr. Patricia Dixon, and then we're going to move over to Dr. Scott Loyler. I wanted to share with you all, too, that as Jenny said, this will be recorded. And we're going to be able to share this link with all of you if you'd like to go over it again. At the end of the call today, we are available to answer some questions as we get towards the last maybe 10 or 15 minutes. But recognize that the National MPS Society has chosen Dr. Patricia Dixon's research as a challenge grant for all of us for mucolipidosis, and we're very excited about that. Dr. Patricia Dixon is the Chief Division of Genetics and Genomic Medicine, Departments of Pediatrics at Washington University School of Medicine in St. Louis. And for those of you that may not know, since the early 2000s, she's researched brain-directed enzyme replacement therapies for MPS. Her research efforts have encompassed MPS 1, 3B, and 3D, with past and ongoing research supported by the National Institutes of Health, the FDA, private foundations, and industry. She has co-organized national workshops on the challenges in developing therapies for brain and inborn errors of metabolism and in the central nervous drug delivery. We're excited now as she's moved research also into mucolipidosis type 2, 2, 3, and 3. The National MPS Society, along with ISMRD, is committed to advancing this research for mucolipidosis, and both organizations are moving forward in support of this novel research entitled Gene Therapy for Mucolipidosis 2 and 3 with the AAV S1, S3 phosphotransferase. And we're going to finish at the end of Dr. Dixon's presentation with Dr. Scott Loyler, who will join and talk a little bit more about our research program and the challenge grant itself. So Dr. Dixon.
Thank you so much, Terry. And thanks, everyone. I want to first dedicate this talk to, of course, all of the families and everything that you have done, but also all of this work stems from Dr. Stuart Kornfeld's laboratory and his discoveries and work, and he's responsible for bringing me into this space to study this further. So let me share this with you, and I think you should be seeing the screen. That's just the slide now. Just in the way of disclosures, I have some research support, M6P Therapeutics, Alnylam Pharmaceutical, and BioMarin, and have consulted for Denali and Mandos, and have a patent for enzyme replacement therapy for Sanfilippo D syndrome, and we'll be talking about experimental therapies today. So when we think about gene therapy and just DNA medicine as a whole, there are different kinds of ways you can think about this. One is what we call a gene therapy or gene replacement therapy, or giving back a gene that's missing. Another is gene editing, trying to repair a gene. There are gene silencing, and then gene activation, taking a gene that's kind of dormant and turning it back on. Today, we're going to talk about gene therapy or gene replacement therapy, where a virus is used to deliver the missing gene. And this virus, we think about viruses as causing illness in our body, but this is not the kind of virus that we use that would cause illness in a body. We replace this DNA, this gene, in a way that's generally permanent. For AAV, which is adeno-associated virus, or one type of gene therapy, and the type we'll be talking about today, that gene stays separately from the patient's own DNA. So Here is a depiction in red here of a cell. And here is the nucleus of the cell, which is where your DNA, your genes live. So here I've drawn some squiggly DNA. They live in chromosomes in the nucleus here. And out here is an AAV particle that comes in, and that gene is delivered and lives there, but separately from the patient's DNA. It will use the same machinery that the cell has to make proteins. And so there it will make the protein from that gene, and then that will be the replacement for that missing protein that's encoded by that gene that's defective. So then you can think about ways of giving gene therapy. There's giving gene therapy into the vein, into an intravenous injection that treats the body. There are some gene therapies that have been looked at experimentally to deliver directly into the brain, which usually requires a neurosurgical procedure, into the spinal fluid surrounding the brain, which can be done through a neurosurgical procedure as well, or into a spinal fluid by lumbar tap. Specific sites, eye, ear, other things, and then newer gene therapy viruses that cross what we call the blood-brain barrier. And what this means is that we have specialized blood vessels in our brain that where it restricts what gets into our brains to protect it. And while a very nice protective mechanism for our brain, it also hinders our ability to get therapeutic molecules that are larger, things like virus and enzyme replacement therapy, into the brain. And so people have looked at different kinds of viruses with what we call the caps, that are the outsides, that are better able to enter into that blood-brain barrier and get into the brain for those conditions where you need to have treatment in the brain, particularly if the condition affects both the brain and the body. So you may or may not be surprised that there are over 2,000 gene therapies in development. This is per the latest Sightline, which is a quarterly report, excuse me, put out by the American Society of Gene and Cell Therapy. 41 gene therapies have been approved globally, and that includes AAV and genetically modified cell therapies, as well as RNA therapies and others. Some of the AAV therapies that have been approved include retinal, or treating the back of the eye for hereditary causes of vision loss, gene therapy for a neurological condition called spinal muscular atrophy, gene therapy for hemophilia, and another neuromuscular disorder called Duchenne muscular dystrophy. So this is a little bit technical, but not necessary to know all of this. AAV stands for adeno-associated virus, a single-stranded, typically DNA virus. And it has the capsid, what I mentioned, on the outside, that determines what we call a serotype that can affect how the virus distributes through the body, what tissues it gets into more or less, and how immunogenic it is, or how much the immune system might react to it. And again, this therapeutic AAV is replication defective, so it doesn't reproduce itself inside the body. It doesn't cause diseases inside the body. So here's an example. I mentioned spinal muscular atrophy. It's a neurological disorder of cells in the spinal cord that control the muscles. There's an infantile form, which is screened for at birth in many states. There's a gene therapy using AAV, which is 9, the serotype, and that's delivered intravenously and accompanied by, say, 30 days of steroids. This is an image from some of the preclinical work looking at how you can see the SMN protein in the cells of the mouse after the gene therapy. So when we think about getting gene therapy into the brain, and if we wanted to do that from an intravenous injection, people have looked at different kinds of serotypes of AAV, so AAV1, 2, 3, et cetera. You can see them here on this graph, and whether they get into the brain when delivered intravenously. And you can see from this graph that if you give a virus intravenously, it gets into the tissues of the body pretty well for many of them, and then maybe some less for muscle and definitely less for brain. But one of the ones that does, at least in mice, get into the brain is AAV9. And here is the graph here. And then on the left, this is a PET scan of mice treated intravenously with, say, AAV7, where you can see it does not get into the brain. You give it intravenously, and AAV9, where it does. Just a little bit of background on mucolipidosis types 2 and 3. These are caused by deficiency of the enzyme UDP N-acetylglucosamine, lysosomal enzyme N-acetylglucosamine 1 phosphotransferase, which Stewart always just called the phosphotransferase, or the PTAs for short. It has alpha and beta subunits that are encoded by one gene, GNPTAB, and that's the gene that's deficient in ML2 and ML3 alpha beta. And then a gamma subunit that's encoded by GNPTG, and that's the gene that's deficient in or defective in ML3 gamma. This is just a little cartoon illustrating the alpha and beta subunits. Just a conceptual cartoon of the PTAs, and then the gamma subunits that bind up here. The alpha and beta subunits start out as together, and then they're cleaved, and that is kind of what the PTAs looks like. It's a transmembrane protein in a part of the cell called the Golgi apparatus. So slide is a little bit busy, but I'll walk you through it. This is just to come up with to understand what that PTAs does and why that's important. The mannose 6-phosphate receptor system is important for how our lysosomal proteins and enzymes get into the lysosome once they're made by a cell. So that PTAs that's defective in ML2 and 3 puts that mannose 6-phosphate or MAN6P on our lysosomal enzymes. And then there are receptors that bind that MAN6P to bring those enzymes or proteins into the lysosome where they can break down the storage material. So that's depicted here in this cartoon. Here's a cell. You have your lysosomal enzymes here that are the squiggles that are made. And then that PTAs puts the mannose 6-phosphate onto those proteins. There's the MAN6P receptor on a lysosome where it can then get in. These lysosomal enzymes also leave the cell, and then they can, when that happens, they can go to a different cell. There are mannose 6-phosphate receptors on the surface of the cell that can bind those mannose 6-phosphate residues and bring it inside. And those mannose 6-phosphate residues on the lysosomes will then pick up the enzyme with its mannose 6-phosphate and bring it there where the storage is. Now, in mucolipidosis, without the PTAs, the lysosomal enzymes lack that M6P. So you can see, again, you have your depiction here of the cell. There's your lysosomal enzymes, and they're not able to successfully traffic to the lysosome through that mannose 6-phosphate receptor, where the storage material then accumulates more pronouncedly. And then the enzymes themselves can leave the cell. They actually leave the cell in greater amounts in ML, not truly understood, but you measure this in the bloodstream with very high levels of these lysosomal enzymes in the bloodstream in people with ML2 and 3. And then these don't traffic well to cells because they don't have that mannose 6-phosphate residue that puts them in through that mannose 6-phosphate receptor on the cell surface, and cannot also therefore reach the lysosomes of those other cells where the storage material is. Now, how might ML be amenable to gene therapy? I don't know how much you follow or hear about other lysosomal storage diseases like MPS and the idea of how those might be treated with with gene therapy. So in those cases, the enzyme that lives in the lysosome is not made by the cell, typically in the patient with the condition. But when gene therapy is given, some of the cells end up with the gene therapy inside, and some of the cells don't end up with the gene therapy inside. And this is because gene therapy cannot treat all of the cells in the body. We're just not that good at being able to get a virus that we give into the vein and have it go absolutely everywhere and get that gene into every cell and fix it. But that's a cell that does get the gene therapy in the case of a lysosomal disease will make the protein, which wouldn't normally be made, and then it will make it in large amounts, and then that protein can then go into the other cell, typically through the mannose-6-phosphate receptor, because that machinery all works. In ML, what we envision is the same thing happening where some of the cells will get the gene therapy, and some of the cells would not get the gene therapy. The cell with the gene therapy will continue to make lysosomal enzymes, but this time they will have the mannose-6-phosphate. And because those enzymes will still get out of the cell that has the gene therapy, just like they did in cells without the gene therapy, those enzymes that do have mannose-6-phosphate will traffic to the cells that do not get the gene therapy through the mannose-6-phosphate receptor so that they can have some therapeutic effect on those cells too. So even though our protein that we have to repair with the gene is in the Golgi apparatus and not a lysosomal enzyme that gets secreted outside the cell, we think that the cells that are treated with gene therapy will make enzymes that are more therapeutic to other cells nearby. So this is another very technical slide, but you don't need to know all about it. But I thought for those of you who wanted to get a little more technical to have that option. This is from Stuart Kornfeld's paper on the S1S3 phosphotransferase. So the wild type, what we call the naturally occurring phosphotransferase, or PTAs, is a pretty big protein of 1,256 amino acids. So that when you try to code for this in a gene therapy construct, it's a pretty big piece of DNA, and it limits a little bit what you can do in terms of packaging and getting it into a virus and getting it expressed. Stuart and his lab developed a shortened form, which is depicted in this diagram, and then down here in this cartoon, of this S1S3 phosphotransferase that's 576 amino acids. It still contains the alpha and beta subunit of the phosphotransferase. It no longer binds the gamma subunit. However, it is able to phosphorylate or put mannose-6-phosphate on lysosomal enzymes with greater efficiency than the naturally occurring phosphotransferase. And so this is a chart from Stuart Kornfeld's lab paper showing cells that are transfected or given the DNA for S1S3, or cells that are not. They're given just the wild type phosphotransferase. And then they're also given the DNA for different lysosomal enzymes to see how well those lysosomal enzymes get mannose-6 phosphorylated in the presence of either wild type or S1S3 phosphotransferase. So this on the y-axis here is the percent of glycan phosphorylation, or the mannose-6 phosphorylation. And the left here is wild type, and the right here is the S1S3 in all of these bar graphs, showing an increase in mannose-6 phosphorylation of these lysosomal enzymes of those cells in S1S3 phosphotransferase versus wild type. We also want to talk a little about, we have an ML2 mouse model. This is also something that Stuart and his group had characterized. There's a couple of papers on this mouse that are cited here, Gelfman 2007 and Idle 2014. On the left from this Idle paper, which concentrates on the neurological impairment in the mouse. You can see up here the graphs of 1 month old. This is rotor-rod behavior, stationary, constant speed, and accelerating speed. And at 4 to 5 months old in the mice at stationary, constant speed, and accelerating rotor-rod. The white here are the ML, the white here are the wild type mice, and the black here are the ML2 mice. Showing that they have less time, which is the y-axis on the rotor-rod, particularly at 4 to 5 months of age, where that's impaired. On the right here, you can see storage material on pathology in the pancreas. So this is a wild type on the left. This is the salivary gland wild type again, and the lysosomal storage material, which is the white. And then here in cartilage, you can see increased storage in the chondrocytes growth plate in the ML2 mice. So we did some proof of principle experiments with the gene therapy with S1S3 in the ML2 mouse model. This graph is showing the mannose-6-phosphate binding of the enzymes that are in the serum of mice that were treated with AAV S1S3. So the black lines and dots on all of these graphs are serum from ML2 mice before they had any gene therapy treatment. And then the other lines and dots on these graphs show the serum from the ML2 mice at 2, 6, 10, or 14 weeks after the gene therapy treatment. The y-axis shows the mannose-6-phosphate receptor binding of different lysosomal enzymes at different amounts of enzyme applied. So you can see all of the mice pre-treatment had very little to no binding to the mannose-6-phosphate receptor of those lysosomal enzymes due to lack of mannose-6-phosphate content on those enzymes. But after treatment, you see an improvement in the mannose-6-phosphate receptor binding, which suggests that they have mannose-6-phosphate now on the lysosomal enzymes measurable circulating in their serum. This graph is interesting to me because it shows that even after treatment with gene therapy, we still see very high levels of secretion of lysosomal enzymes in the ML2 mice compared to what we normally see in patients who don't have ML2. So this graph on the y-axis again is the phosphorylated enzyme. So these are enzymes that have mannose-6-phosphate, so mannose-6-phosphorylated lysosomal enzyme content. The black bars here are all of the ML2 pre-treatment serum, so no mannose-6-phosphate. The gray here, very hard, maybe a little faint, but that's the normal amount of phosphorylated circulating lysosomal enzymes in mice that do not have ML. So what we would expect to see without ML is really not very much circulating lysosomal enzymes with mannose-6-phosphate content. However, after gene therapy, we see elevated levels of not just of lysosomal enzymes, but elevated levels of lysosomal enzymes containing mannose-6-phosphate. So this is encouraging that potentially the gene therapy that's treating some of the cells, that those cells are continuing to make high levels of enzymes, and those enzymes have mannose-6-phosphate on them, which is encouraging that they could then cross-correct their neighbors. We looked at S1S3 expression in the treated mice. We could see good expression in liver, heart, not quite as much in brain in our first experiments here. And then in liver, 50 weeks after dose, we saw by staining for the gene, we can see good expression in liver and brown adipose, still even up to a year, which is as long as we studied so far. To showing that there's good duration of the gene therapy in the mice. This is a picture of the storage material. So this is a salivary gland on the top row, another salivary gland, which is the parotid gland, that's the name for it, and then the pancreas down here. And wild types are the column on the left, the center column are the untreated ML2 mice, and then on the right are the gene therapy treated mice. So you can still see some storage in the gene therapy treated mice, and but it appears to be less in some cases than the untreated ML2 mice. We didn't see substantial changes or improvements in length, weight, and bone mineral density with the treatment with the gene therapy in these experiments. Now, these mice were treated at age 4 weeks, which was so not at birth, and it is an ML2 model, so it's possible that that is part of why this happened, but we didn't see any improvement in these early experiments. We did see an improvement in survival. These were the ML2 mice here that were not treated. Wild type and heterozygous mice were similar with a survival. You can see this is age in weeks. So 1 year would be about to here. This is 80 weeks, 100 weeks. So we'll have about 2 years. So there was a 12-week increase in survival in these mice, 3 months. So from these initial experiments, we were encouraged that the gene therapy is able to put mannose-6-phosphate back on lysosomal enzymes. We saw some impact on the ML2 phenotype in these mice. They were adults. At 4 weeks, their mice are considered adults when they were treated. So it's possible that earlier on treatment in this knockout mouse would have been more effective. We saw long-term expression and partially improved pathology. So for next steps, what we'd like to think about is, can we further improve efficacy? So we plan to test different gene therapy designs, AAV gene therapy designs, in different technical ways. So one way is to, for example, change the promoter so that we are expressing in each cell potentially a little less amount, but maybe that will allow us to push the dose a little bit higher so we get more cells transfected. Can our results in mouse be replicated in another model? We have only looked at this mouse model. It does have a lot of similarity with ML2 patients, but it is not a perfect model. For example, the ML2 mouse has substantial retinal degeneration and vision loss that really isn't part of mucolipidosis in people. So we plan to test the gene therapy in a larger animal model. Will gene therapy with S1S3 improve different patient mutations? This protein, as you saw from the cartoon, is a little bit different than the wild type, whereas patient mutations might interact with it a little bit differently. We don't think this will happen, but we want to test the response in different patient cell lines to be sure. And then finally, just a very exploratory work to see if potentially S1S3 would also phosphorylate lysosomal enzymes for patients with ML3 gamma, because it does not require gamma for binding. It doesn't bind to gamma, and it's possible that it could phosphorylate the enzymes in the absence of gamma. So we plan to test the response in an ML3 patient gamma patient cell line to see if that is the case. And so I think that's the end there. I'll stop sharing if I can find the button. And I don't know if you wanted me to take questions now or if we're going to do that at the end after Scott.
Yeah, I think we can take questions now. If anybody has questions, put them into the chat. I know that Jennifer had gotten some questions already, so she's going to ask you some things, Patty, directly.
Okay, great.
Yeah. Sorry, guys. Yes. So the first one that I have is, with this current gene therapy, how is it planning to be administered for mucolipidosis?
We're hoping to be able to deliver this intravenously only, that would be the route of administration. I didn't mention, but we would like to think about whether so AAV9 is great in mice. It can cross into the blood-brain barrier. People are doing more experiments with other types of gene therapy that might cross a little bit better. If we have the ability to explore that as well, I think that could be very useful. But we're hoping to deliver this intravenously and not need any additional areas that are injected.
Okay, thank you. And then the next question I have is, what preclinical development still needs to happen in order to move this into patients?
Yeah, so what we need first, we want to explore more proof of concept just to make sure that we have chosen the correct and best design for our virus. If there are some that could work more, so we can optimize efficacy, I think that we would need to explore that first, just to either prove that this is the best that we can get, or to find something that might work a little bit better. After that, we plan to approach the FDA to see if we can get some feedback into our design. They have laid out the plan for what gene therapy should be doing to get approved by the FDA. And so we would follow the prescribed toxicity and distribution studies and such, and hope that they will give us some extra feedback on what those are. But they have laid those out. Does that answer the question?
I think, yes, I think that answers the question, unless anyone else wants to chime in. Okay. And then finally, do you have an estimated timeline at this point to get to a clinical trial or get this into patients somehow?
So a lot of that is, of course, up to regulatory agencies and how they react to our data. We we think that the preclinical work that we have planned is doable in a couple of years. I wish I could make it shorter, but the mouse model and also the larger animal model work does take some time. Even the mouse model takes a year to show survival endpoints, and months to show pathology endpoints. So I think it will take that full time to get all of the studies that we need to make a good package to get to the next stage, which is all the pre-IND studies that we would need to move forward.
And then I did have a question come through the chat. Well, I'm trying to enlarge it. Is there a mouse model for ML type 3, and do we need one? And if we don't have one, how do we get one?
Yeah, I'm not aware of a mouse model for 3 alpha beta. There's a 3 gamma mouse that we could study if that turns out to show some promise in ML in the patient cells. We also have a NIM mouse model that is a another mutation for ML. We have that model in our lab and breeding, so we can test if it's helpful and in another model to see if we get similar results.
Would the ML2 model, could we apply that to ML3, or are we going to need to develop an ML3 model at some point?
Um, I don't think that we would need to develop an ML3 model. The problem with the ML3 model is that the phenotype is going to be even more
Slower?
attenuated and slower progressing. So, it would be good for finding out if you can phosphorylate the enzymes in the mouse model. But I think we can answer those same questions in patient cell lines, and maybe even better, because you can more easily and more readily test different mutations with the patient cell lines. We can measure the output in terms of the Mana6 phosphorylated enzymes. So my hope is that we can address all of those questions about mutations in cell lines and just leave the animal model as to more of questions about distribution and then overall impact on, are we reaching some of those target tissues?
Okay. And then,
The enzyme, yeah, sorry, go ahead.
no, go on, go on.
It popped up and I started immediately answering it, but maybe you have a different question you want to pull up.
No, go for it. Go ahead and answer whatever
Um,
you said.
okay, the one that popped up was how often was this administered? So the mouse model we treated with gene therapy, which was treated one time. We did another test where we gave an intraventricular dose to the newborn mice, and then 4 weeks later gave a dose of intravenous gene therapy. We can do that in the mice because we did a special test to make sure they hadn't developed antibodies to the virus. It's kind of a proof of concept trick we did. But generally speaking, right now gene therapy is hoped to be one and done. And it wasn't it was not like an enzyme where it's given over and over. Um,
And then I want to jump in real quick, and then we can go down the rest of the list of questions. Since you are thinking patient cell lines is probably the best way to go to test this, can you tell us a little bit about the process for getting patient cell lines and what is required?
Yeah, so we have a study, it's open now called the BOLD study or Basic Observations in Lysosomal Diseases. This allows us to collect blood from individuals with lysosomal diseases, including ML. And we have a wonderful genome engineering and stem cell core here at Washington University that can program from blood iPS cells. And then from those iPS cells, we could generate any number of different kinds of cells to study. We also could collect patient fibroblasts for skin biopsy or from we can accept samples from outside as well. For a lot of these, there are readily available commercial cell lines at Coriell that can be a place to start, which is I believe where we're starting now. But we do have a study coordinator who is for our BOLD study. If anyone is interested in participating, I'd be happy to discuss that with you. It would be very helpful.
Thank you. And going back to the list of questions that I have, which large animal will you be looking at to move into next?
So this is a feline model that's at the University of Pennsylvania at the vet school that we will be studying.
And going
Very
back to this,
limited, yeah, very limited study, as small numbers as we can for this study.
and going back to the cell lines real quick, if there is a link for the study, someone is asking how we get in touch with the BOLD study.
Oh yeah, well, let me work on that while Scott's giving his talk, and I'll pull that up and put that into the chat for people.
Okay. And then just a couple more questions for you, unless they keep piling in, then we can move over to Scott. Is there an effort to collect run-in data during this time?
We've had a number of discussions about this. I think we're still not we're we're still thinking about the best way to go about that. But yes, I think the short answer is yes, I think that would be a good idea. And we would appreciate any guidance on, what would be the best way to do that at this time. We are, I think a lot of it too, if we can get some feedback from the FDA as to what might be acceptable for if accelerated approval pathway would be potentially acceptable, or what other kind of data, if not, would be best. And I think as we go forward and kind of see what indications this seems to work best for, we will have a more refined answer to which population we'll be needing to address next.
And then Hannah, I might leave your second question for after Scott's talk because I think he can answer where we're at with funding for this research. So Scott, I will hand it back off to you.
All right. Thank you,
There is one
Jenny.
more question that Dr. Brock put in. Did you take a look at that? This week,
Oh,
a
yeah.
report published in Nature Medicine on contaminating plasmid sequences and disrupted AAV vector genomes leading to liver toxicity after a few weeks. Gene therapy, have you looked in your treated mice for these harmful vector-human fusion junctions, Dr. Dixon?
Yeah, we haven't done any safety evaluations yet. I think right now we're really at the stage of optimization and efficacy vectors. I definitely, as we think about new vector designs and whether they might be better and more efficacious, we also are taking safety into account and what's known about what the, most recent literature says about safety. So, I know it's Mark Sands, obviously at WashU, was my colleague for years before he recently retired, and in the office next to mine, and he was the one who discovered in 2005 that mice with gene therapy can get hepatocellular tumors, even if AAV is given, which was supposed to be very safe. I think we're learning a little bit about what factors might increase the likelihood of this. It's certainly something we're going to look at very carefully as we move forward, and and just be very mindful of this.
And then we do have two more questions about age. So if you have an inkling, Dr. Dixon, will there be an age maximum for the treatment? And do you have an idea if age will play some role in this gene therapy?
I mean, if the question is about the clinical trial, we don't have the clinical trial design at this stage. So I don't know the answer to that. If the question is about what patient age might be amenable to treatment, at this time it would be similar to other conditions, with gene therapy, that it's not necessarily with AAV gene therapy, it's not necessarily an age restriction based on who can be treated with it. There are, as with all of these conditions, as we know, they tend to be progressive, such that we know that earlier administration will have more of an impact. We're hoping that we can treat symptomatic disease. Obviously, we know that that's the most important thing to so many of you, and not. I would prefer to not have have restrictions in any way, but we just don't know yet. I just can't answer that question.
Thank you, Dr. Dixon. I really appreciate your presentation today and really delivering it to us in a way that help us even more solidly understand the gene therapy process, for mucolipidosis. And certainly we've seen this across other lysosomal diseases and how it actually looks. And so there can never be enough times that we have this. Again, for everyone, just so you know, this is recorded, so you'll be provided the link to go back and take a look at it. If you have any other questions, again, you can follow up with Terry at mpssociety.org. You can follow up with, is it Jenny? Jenny, can you share your email address?
Yep, it is Jenny at ISMRD.org. I can put that in the chat as well.
Thank you. I appreciate that. We'd like to transition this because this is really quite an exciting point of inflection for mucolipidosis when we see research moving in this way. And certainly many of us that are on the call, and I know many of you have waited decades for a moment like this, but it isn't going to go forward unless we can find the funding for Dr. Dixon's work. And so the second part of this call really is about how do we rally the troops, and how do we rally the troops in order to begin funding for the challenge grant that Scott's going to talk to you about. So Scott, I'm going to go ahead and pass this over to you.
Thanks, Terry, and thank you, Patty. This is a great presentation, and we're really excited about getting the funding for your treatments to go forward and get them tested in people as soon as we can. I'll reiterate, this is a historic challenge grant for you. We're hoping it's the first ever gene therapy for mucolipidosis type 2-3, which is an ultra-rare fatal disorder with no approved treatments. Our goal is to raise $500,000 by the end of 2027. This is critical for your funding to get this proof of concept completed and tested in a large animal model so we can move forward with the IND filing. So an IND, investigational new drug application with the FDA, That's the first step in getting this tested in humans. And it's critical that we get everything done and completed as quickly as we can. I think you were a little, what's the word I'm looking for? You weren't very pronounced in the S1S3 phosphatase. It's a remarkable enzyme that Stewart developed that's, from what I can see, 20 times more effective than the wild type enzyme. So it really will make a huge difference for this disease, we hope. And you have the ability to use that from the patents issued by WashU. So that's going to be a real breakthrough for this gene therapy, and that's why we're moving full steam ahead with your program. We typically only fund $100,000 grants, and this will be a first for the society, funding a $500,000 grant. But we have to raise the money now to get all of this data generated and get everything needed for the IND submission. So that's a heavy lift. We've just started the fundraising now, and we hope to continue to raise that 500,000 quickly, and then more money is going to be needed to get it through into a clinical study. So we recognize that, and this is just the first step towards that goal. And it's time now to rally everyone we can to get funding for your program and move it as quickly as we can. I can talk a little bit about the safety of AAV. That was brought up previously. I've done AAV gene therapy research myself for more than 30 years. I know it's a very safe and effective vector. I was part of the early clinical research that was done for the spinal muscular atrophy program, and that's a program where we put more AAV gene therapy into the smallest paid babies we could, and those babies are doing amazingly well. We've not seen any tumor development on those kids. It's a very safe and effective treatment. But we have to use a lot of vector. So that's why one of the goals for Patty's program is to optimize the system so that we can use even less vector to have a greater benefit. Some of the studies that Patty mentioned, done by Mark Sands, that was work done in a mouse model, and those mice don't contain, or humans don't contain the same gene region where the vector integrated causing tumors. So we don't see the same effect in humans. So we think that was more of an artifact in the mice. And even though we see some integration events over time in human cells, we're getting much better at purifying the viruses during the manufacturing. So it's much cleaner preps today than were used even 5 years ago. We're getting much, much better at that purification. And the FDA is getting much keener on what are the requirements for giving a safe and effective gene therapy vector. So we're really excited about Patty's program, and we want to move it along as quickly as we can because we think it's the best option for these kids. Any other questions?
Scott, yeah,
Yeah.
I have one just from Hannah from earlier. Scott, if you don't mind telling us where we're at with funding for this research now, or Terry.
Yeah, well, we're just beginning. I heard we have gotten a few donations thus far. I don't know offhand what the total amount is yet, but we have started.
We
Terry,
have started. Yeah, I'll kind of kick in here too. Thanks, Scott, for providing some context behind it. I think one of the important statements that you had shared with us is, well, a couple of things to summarize and then go to funding is as Dr. Dixon shared, we're in right now studies, and we're looking at best ways to optimize administration. And the first 500,000 trench, I call it, or tranche, I call it, is towards those studies. And as soon as we can get that funding, and the sooner we can get that funding and ensure that Dr. Dixon has those studies can begin the way we need them to in a rapid way. And so fundraising is critical right now from the entire community and for all those that care about mucolipidosis. And then the other piece was when you had talked about the fact that you had recognized that this S1S3 has 20 times more, I guess the word for me is the layman, maybe a potency potential in terms of delivery. And then the third piece was, please recognize what Dr. Dixon shared in terms of the administration happened with a mouse model that was classified as an adult mouse model. And so that's the data that she's sharing there in the outcomes and the graphs. And so imagine now if we're able to pull that back in her studies and her lab to mice that are younger, because we all know that when we treat someone that's younger, it's going to have the optimum outcomes as well. But we are in a fundraising stage right now. I can share with you that there's going to be multiple follow-ups for all of you on the National MPS Society and the ISMRD side in terms of what it's like to be able to bring funding through our mechanisms to support the challenge grant, to support Dr. Dixon's work at the lab. I want you to know that we need each of you. We really do need each of you. And we need your networks as well, because we've gone now 3, 4, and 5 decades for some of you who are on the phone. I mean, on the Zoom call right now. I know how long you've been in this community, but we're here. We're at a moment that we have ability to make change. And as many of you know me, and if you've known my daughter, Jennifer, and you've known the community, when can you share that we've been able to outreach at this level before to say we're on to something? Well, folks, we're on to something, and we're on to something now. And that's going to require this incredible amount of funding that we need to reach for Dr. Dixon's lab. And then also recognize the fact that we know that's just the beginning of the tranche. Okay, it's just the beginning. We know that there's more that needs to happen. So we're reaching out on a global level, which is why I appreciate anybody who is in the United States West Coast time, as I see Kevin Gates or others that came in early in the morning. Please note that we were trying to reach the best time frame on a global level for everyone and be sensitive to their times. Information will follow quickly, but always note that we will have something available immediately on our website where you can donate now, and you can click Mucolipidosis ML Challenge Grant. That's one way. Some of the most recent ways that we've been raising funding for research with families on our side, Jennifer, is we have courage pages. And that has gone really, really well because families have had an opportunity to tell their story. And through that story link, it's then been utilized in their networks around the world. And we've raised so much funding for research just through storytelling, where you're sharing with your friends and your families about your child, about the disease, about Dr. Dixon's work. We can provide you these truncated talking points that we want to give to you that you can include in your story, and then share photos and photos and photos and tell everyone the importance of bringing funding across the finish line. I also know with Jennifer at ISMRD, they'll also have a link too that they will be able to bring funding in that way as well. Their board of directors is fully supportive of the work of Dr. Dixon, and we couldn't be more thrilled to have this partnership. But immediately following this meeting today, we're going to send out to you some more information because I've already put the email together and I'm going to get it out to all of you right away. But we need you. We're not going to be able to do this on our own, and it's going to take all of us to do this lift. And I really appreciate you thinking that way. I appreciate you thinking about your networks and how to reach to your family and friends, because it's time, you guys. There's something here, and it's time for us to link onto it. So thank you, Dr. Dixon. Thank you, Scott. Thank you, Jenny. Does anybody else have anything else they'd like to share right now before we call it?
Perry, I'm just going to add on behalf of the Board of Directors of the National MPS Society that we are fully behind this effort. This has been a long time coming. We know within our family of diseases, we've had the haves and the have-nots for way too long. And just we're excited and want to collaborate with all the other disease groups out there that we can all work together and make this happen.
Thanks, Steve. I really appreciate those comments. Jenny, any last things you'd like to say?
No, I think there's multiple opportunities. So as Terry mentioned, you can donate directly to the MPS Society and designate funding towards ML research. We are revamping ISMRD's donation page as well. So you're going to have the same opportunity to do that on ISMRD's page. And I would also, possibly recommend starting a fundraiser where you can reach multiple people within your community. I'm going to jumpstart my own fundraising campaign with the goal of raising $111,000 this year. I know that's a lofty goal, but this is really important to me and my family. So I would recommend you guys all think about that and see how we can get to the $500,000 goal as soon as possible.
Well, thank you, everyone. I hope you have a wonderful Sunday or Monday, depending on where you are in the world. And I will follow up with you here in the next 30 minutes with an email and a link to this recording. Thank you, everyone. Bye.
Thank you all.